I have to start by saying that this has been a great collaboration with our colleagues at the Structural Genomics Consortium. And a lot of the work that we've done initially on many of these targets have been in open science forum. And then last few years, we've also joined the UB Open, another great collaboration, which also the SGC plays a very important role in. So with that in mind, this family consists of over 260 proteins. Many of them have been solved in terms of X-ray crystal structure, and many of them have been solved by the SGC. So it made perfect sense for us to collaborate together, given the SGC's strength in that area. And beyond their 3D shape, there's low sequence conservation among these family members. Therefore, when we think about maybe drugging some of them, potentially selectivity is not a major issue as it would be in the case of kinases, for example. But more importantly, these proteins function as scaffolding proteins in many complexes in diverse cellular processes. So we felt it was a great opportunity for us to further explore and better understand the biology of these targets by developing small chemical probes to them. Then we can make available to the community so that additional biology can be undertaken so that we can better understand their potential as therapeutic targets. So, but also, as you all know, we're focusing on cancer, and we know from much of the literature that they have great potential in cancer, but also in other diseases. So the team is going to tell you a little bit about two targets, but we've looked at many other ones, and these are not always trivial to drug, I must say. We've explored many with the SGC, and of course, we're going to tell you about the successes. But I have to say, there are many where we have not been able to find a good starting point chemically. That is for us chemists to optimize. So this family is unique in that one of their features is a short structural motif about 40 amino acids, and often terminating in W and D dipeptides, hence the name. Okay, and they adopt this sort of propeller type, donut type shape, and so that's a feature, for example, in for sure WDR5, which is only WD domain. But then a protein such as DCAF1, as you can see, is far more complex. And as I mentioned, you'll hear more about the details, so I will not dwell on that. But with that, I also want to move on to our first speaker. So I'm going to stop sharing. And also highlight the fact that this effort here also highlights our collaboration between the drug discovery team and group and the bioinformatics group at OICR. Okay. So our first speaker, Sam Majoros, is gonna present our bioinformatics effort in this area. And she works very closely with Dr. Quan Tran, a member of Dr. Lincoln-Stein's group. So this is really a highlight of the fact that I'm very happy about that we're collaborating within OICR and especially with our talented bioinformatics team. So Sam is a bioinformatician, and she graduated with her Master of Science in Bioinformatics from the University of Guelph before joining OICR. She is a research assistant at the university. She was a research assistant at the University of Guelph when she specialized in the fields of ecology and evolutionary biology. And Sam leads several collaborative projects and uses her computational skills to provide support for the many projects in the drug discovery program. So I'm going to turn it over to her to tell you about her effort around the WDs.
Okay. Well, thank you so much for the introduction. So my talk today discusses some of the bioinformatics work that's being done to support the WDR projects, as well as the general process of taking the wide range of data we have available to us and filtering this down to genes or cancer types of interest that we can then investigate farther. Oh, okay. So this is just an overview of some of the things we're doing as part of the bioinformatics work. So it really all begins with the data. There's lots of data available from databases such as cBioportal, DepMap, Reactome, and String. This data includes expression, mutation, copy number, and pathway information for over 20,000 genes across cancer types. And we want to use this data to identify biomarker candidates. So in the figure here, there's some examples of some of the data we work with or some of the analysis you might do, things like co-expression, mutation rates, copy number variations, differential expression, tissue specificity. And one of our goals is to develop a pipeline and series of analysis to narrow down genes of interest for a given cancer type or to determine a cancer type of interest for a set of genes. And I'll go ahead and go through some examples of workflows and steps and analyses that we will do during this presentation. And then another goal we have is to develop an online data portal that allows researchers to easily interact with the data. So this would be that integrated knowledge database there. So I've been working to bring together data from all of these sources, pre-process it, and make it available through an interactive user interface. And I'll go into more details about that later as well. So in a future goal we have is to implement machine learning to give genes a target prioritization score. A machine learning model could use all of the data we pre-process to more efficiently investigate each gene and determine if it's something we should look into farther. I won't really talk about that too much today because that's still in the very early planning stages. So today I'll just talk about the workflow, data curation, and the data portal. So the goal of our bioinformatics work is to take all of this data and filter it down to areas of interest through a series of analyses. So these are some example workflows. So it does not have to be a linear process. Steps and analyses can be done based on the needs of the researcher. These are just some examples. So for example, you might begin with a cancer type of interest and you want to get a list of genes that you want to look into farther. Or you might have a gene list of interest and want to know what cancer type would be best to focus on for that gene list. And so I'll go through some examples. So if we're starting with a cancer type, we're going to use colon cancer as an example. We might want to begin by getting a list of top differentially expressed and tumor-specific genes for that cancer type. So we might begin by looking at tissue specificity. So during this analysis, genes are given a tissue specificity score based on their mean expression or standard deviation. I then took the top 20 genes and plotted them using the TCGA mRNA expression data. So that's the plot we can see on the side. So the expression is the x-axis, cancer types are the y, and we're looking for a gene that is expressed more in a specific cancer type. So the gene shown here is NOX1, which came up as one of our top 20 genes. And we also see that in the literature, it's reported as being overexpressed in colon cancer. So this kind of validates the scores, the tissue specificity scores we're giving and the analysis we're doing, that we are able to find results that are then supported by the literature. So then we might take those top 20 genes we got and want to look at their pathways. So pathways can be found for each gene using data from Reactome, and we're looking to see if those highly expressed genes are focused in any particular pathway. So here we can see there's a number of genes along the x-axis, pathways along the y, and out of those top 20 genes, majority are found in signal transduction, metabolism, protein, and disease, so spread across a couple of pathways. And if at any point in the analysis, nothing of, or in the workflow, nothing of interest is being really found, and you might need to expand that gene list to then look at things farther. So instead of using top 20, you could do top 100 genes, or do things like look at the interacting partners of those genes, or look for highly correlated genes. So I'm going to jump over to our kind of other example. So what if we started with a gene list? And so for this example, I'm going to use the ribosome WDR gene, so including WDR12 and WDR55. And we're going to begin by looking at the differential expression of those across different cancer types. But just a little bit of background on WDR12 and WDR55. So they're both part of ribosome biogenesis and are part of the large subunit. The WDR12 project was started by PhD student GSUP and based on depth expression analysis. And the current status of these projects, there are publication manuscripts on WDR12 and WDR55 in preparation. And WDR55 is with Dr. Dahlia Barsight-Lovejoy at U of T. And there's also a few potent WDR12 and WDR55 tool compounds that have been developed along with SGC. So we're going to use WDR12 as an example, but we can look at the differential expression between tumor and normal mRNA expression across different cancer types. And again, this data is from cBioportal. So each of these boxes is a cancer type, and the y-axis is the mRNA expression. We have the tumor in blue, the normal is the orangey pink colored one. And we're looking for cancer types where the tumor is there's significantly more expression in the tumor than there is in the normal. And we can see that across several cancer types. The ones with the highest p-values are highlighted. So we can see that WR12 is overexpressed in various cancer types, including breast cancer, colon cancer, and lung cancer. And we can also look into expression farther using heat maps. I'm just going to go through a simplified example of a heat map first before getting into the real data, just so everyone knows kind of how to read it and what's going on before I add more information. So the rows are the genes, the columns are the patient samples, and these are clustered together based on their mRNA expression. So genes that have similar expression will be clustered closer together. Patient samples that have similar expression will be clustered closer together as well. And then in the middle is the mRNA expression, so red if it's higher, blue if it's lower. And then there's also an annotation along the top saying whether each of those samples is a normal sample in the pink or a tumor sample in the blue. And we're looking for genes where the expression is lower in normal and then higher in tumor. So this is our ribosome WDRs in breast cancer, which was one of the cancer types where there was significantly higher expression in the tumor compared to normal. And by plotting it in the heat map, we can look and see if there is any patterns between the breast cancer subtypes. Is there higher expression in a specific subtype when compared to normal or other subtypes? So it's very similar to our example one. There's just a couple extra annotations. So there is stage, sex, and subtype. And it's grouped together by subtype just to allow easier comparison. And then there is also an annotation that shows whether each sample has a TP53 mutation or not. And this is, again, data from TCGA. And there's also an annotation along the side just saying whether each gene is part of the large or small subunit, and also marking those control genes because we did include some breast cancer control genes in the heat map. So a lot of the ribosome WDRs are highly expressed in the basal subtype of breast cancer, as we can see with WDR12. It is showing overexpression in this subtype when compared to normal and also compared to the other subtypes as well. And it's like that for a couple of the ribosome WDRs. But if we look at WDR55, we're actually seeing a very different pattern than WDR12, even though we might expect them to be similar. So WDR55 has very low expression in the basal as well as the normal. There's some areas of higher expression in the other subtypes, but if we focus on the basal subtype, we can see that WDR12 and WDR55 are almost showing an opposite pattern, which is interesting. So by looking at the subtypes, we can see that for WDR12, the basal subtype of breast cancer could be an area of interest to focus on farther. So some other things we can look at, whether you're looking at a gene list of interest or a cancer type, you can look into things like mutations and copy number. So this is the mutation frequency. Again, we're sticking with the ribosome WDR example, and this also includes the control genes from the heat map as well. So this is just everything that was with the heat map. But these are the mutation frequency for all of those genes. So that's along the x-axis, genes along the y. And we can see there's very low mutation frequency for majority of the ribosome WDRs. And then genes like T53 have a high mutation frequency, which aligns with what is known in the literature, with TP53 mutations being very common in breast cancer and missense mutations being the most common. Also, TP53 missense mutations result in higher expression sometimes of TP53, which we saw in our heat map as well. So we can also look at copy number. So this heat map is very similar to the one I showed you before, but it's no longer showing mRNA expression. This time it is showing copy number. So if there is a gain, it is shown in red. If there is a loss, it is shown in blue, and then white is just no change. And there is also an additional annotation along the rows that says what chromosome each of these genes is on. And so based on the literature, the most reoccurring amplified regions were on chromosome 8 for triple negative breast cancer samples. So that's things like MYC, DCAF13, and BOP1. And we can see in our data, there is that red area showing the gains. And then the chromosome 5 had some of the most reoccurrent deleted regions. That's things like WDR55, WDR36, UTP15. And we can see as well in our data that those are areas with the blue with the losses shown. But the paper also found that there's not a strong correlation between mRNA expression and copy number variation, which we saw as well. So just because a gene has a gain doesn't necessarily mean it has higher expression. So one last thing we can look at is the depth map expression. So we don't just want to look at the patient samples, but also at the cell line. So this is the depth map expression of our ribosome WDRs for the breast cancer cell lines. And so we can see that some cell lines are more expressed than others, so showing that red compared to the blue. And we can see that for WDR12. Oops, I wanted to go back. Here we go. And we can see that WDR55 and WDR12 still don't show the same pattern all the time. So we can see that in these cell lines where there is higher expression in WDR12, we can see there's still blue and white and a little bit lower expression seen in the WDR55. So by using bioinformatics methods to filter through the data, we can efficiently determine areas of interest to then pursue farther. We do plan to make generalizable scripts for each of these steps, so these analyses can be quickly applied to any kind of data. And we also plan to make these scripts publicly available to other researchers. So through this workflow, we can move projects forward and determine areas to focus on, as is the case of the WDR12 and the ribosome WDRs, where an area of potential focus could be that basal subtype of breast cancer. And the last thing I'm going to talk about is the data portal. So up to this point, a researcher might send me a list of genes they want information for, and I'll generate a heat map or run analyses and then send that back to them. And then maybe they'll refine that gene list, send me a new one, and it's kind of a back and forth of information. But the data portal is one way that can make this a little bit more efficient. So we obtained and pre-processed data, including mRNA expression, mutations, gene correlations, and protein-protein interactions from databases such as cBioportal, String, and DepMap for all genes across 32 cancer types. And in collaboration with Dr. Melanie Courteau's group from Adaptive Oncology, we developed an internal data portal with a web interface using the Overture Portal UI. So this portal allows researchers to access data for any gene and cancer type. So a researcher could go onto the portal, say, I want to look at WDR12 or WDR5 or any gene and pull up things like expression or mutation for that gene within any cancer type. And this is a screenshot of what the portal kind of looks like. So in the middle, you would have all of your data. And then this is sortable columns. You can also adjust what columns you see, so you can get just the information you're interested in. There's also options to download that data and do further analyses with that. And then along the side, there are the filters. So you could select specifically what cancer type you want to see, what gene you want to see. And there's also things like adjustable sliders. So this is a correlation table, for example. So you could adjust the Pearson correlation and say, I only want genes that are correlated over 0.8. And then if you apply that filter, it would affect what you see in the table, and you could look at that information that way. We're also working on cross-table filtering. So if you applied filters to this table and you got a new list of like highly correlated genes, you could then take that gene list and automatically apply it to the expression table or mutation table and pull up information for those genes. And we're still working on improving functionality and adding to this all the time. So some next steps for some of the bioinformatics work. So for the workflow, we want to make the scripts generalizable for each step and make the scripts publicly available to other researchers. For the data portal, I would like to continue to add more data, including different data types and different data sources. Also continue to work on the interface and add in a dashboard and visualizations so that when you go to the data portal, there will not only be a table, but also things like bar graphs and figures and visualizations just to make interacting and understanding that data very easy. And then a future goal would be the machine learning, which again is still very early planning process, but the goal would be to eventually develop a preliminary machine learning model and determine which model and parameters are best suited to our data, and then to test that model on known biomarker genes. So some takeaways, the data portal is a useful tool that will make data more accessible to researchers and improve the efficiency of scientific research and drug discovery. And by going through the bioinformatics workflow, we can determine genes of interest and find target patient groups for genes and compounds, such as in our example, where that breast cancer basal subtype could be a potential area of focus for WDR12. In conclusion, bioinformatics work provides vital information that helps decision making and supports the next steps in the drug discovery process. So thank you so much for listening, and I'm happy to answer any questions during our question period.
Thank you, Sam. Great. And now we can move on to our next speaker, Brian Wilson, who I've had the privilege of working very closely with while at OICR. So Brian graduated with a Master of Science in Synthetic Organic Chemistry from Queen's University in Kingston, and went down south and spent about 9 years as a medicinal chemist at GlaxoSmithKline in the Research Triangle Park in North Carolina. And we were very fortunate to recruit Brian back to Canada, and he's been at OICR since 2009. And he's made many contributions to a lot of our many different projects, but specifically within the WDR family. And he's worked on WDR5 and decaf, and he's going to tell you a little bit about his effort in these areas. So, Brian?
Okay. So as Rima mentioned, I've spent a significant portion of my time here at OICR working in the space of the WDR family of proteins. And today I'm going to talk about two of them, decaf-1 and WDR5. Both have been worked on separately, but I'm also going to talk about the application of one in the space of the other through targeted protein degradation, which is kind of a hot topic in drug discovery these days. So in terms of targeted protein degradation, I think the first thing that we need to take a look at on trying to understand, and this is a very, very simplistic cartoon that is for illustrative purpose only to kind of show the general nuts and bolts of how this works. But cells need to regulate proteins so that they can function properly. There could be various reasons why proteins need to be removed from the cell. Either the expression level is too high, or maybe there's some adverse event occurred and the protein is misfolded. And one of the major ways in which a cell can regulate the proteins that are present is through the ubiquitin proteasome system, the UPS, which I've shown here in this cartoon. It essentially breaks down into a process where cells use three enzymes, conveniently named E1, E2, and E3, in which they transfer a protein called ubiquitin onto a particular protein substrate. And then when you have these ubiquitins kind of tagged onto your protein, it flags them. And then that protein is recognized by the proteasome, which can then essentially digest that protein, break it down into its component parts, either subunits of protein, smaller amino acid sequences or individual amino acids, which the cell can then recycle for other purposes, and then re-release the ubiquitin, which can then re-enter the cycle and go on to its next task. We do know a fair amount about these E1s, E2s, and E3s, and you can see here in the bullet points on the right, there are many, many less E1s and E2s than there are E3s. And the relatively large number of E3 ligases provides the required high specificity in targeting vast numbers of proteins that need to be regulated through this process. So each individual E3 may be responsible for the regulation of only one or two proteins. If you only had one or two E3s, you can imagine that would really dominate everything in the cell, and you could end up with a situation where you get very drastic outcomes if that system gets out of control. So it's a level of control by having so many E3s. One of those E3s is one of the WDR family members that I'm going to talk today, DCAF1. You can see DCAF1 is a nice, easy way to say this mouthful right here. And it's a substrate recruitment module for two E3 ligases, the ring type and the hex type. As Rima showed earlier, it has a very traditional WD shape component to it with this 40 amino acid 7-bladed propeller or donut section. And in the field of virology, HIV has been shown to take advantage of DCAF1 using a lentiviral accessory protein, VPR, which binds to DCAF1, which then recruits another protein called SAMHD1, which is a cellular restriction factor. And then SAMHD1 gets degraded, and then that allows the virus to replicate more freely without those restriction factors governing the cell. DCAF1 has been associated with many processes within the cell, and in terms of a medicinal chemist in the field of cancer therapeutics, cell proliferation and tumorigenesis, the role there makes it an interesting target for us to target to try to shut down and create a drug that can stop proliferation. So we set out to do exactly that with our colleagues at the SGC. I meant to mention that on the first slide and forgot. All of the work that I describe here today was done with our collaborators and colleagues at the SGC, both for the DCAF1 and the WDR5, and it was done in an open science manner. We came up with, we started looking for hits using what's called DEL-ML, which is a DNA encoded library machine learning protocol, where libraries of 58 billion compounds that had these DNA tags on them that tells you what the structure is, were screened against DCAF1, and then you can enrich those binders and sequence the DNA to identify the binder that was tagged by that DNA. And then you can use all of that information to build a model of what a good DCAF1 inhibitor would look like. And then you can use that model and you can apply that model onto libraries of commercial compounds that are available for purchase. So this process was done with our collaborators, and we came back with 33 predictions that were purchased and tested. This resulted in the compound shown on the right, and I will say for the non-chemists in the crowd, I have very limited chemical structures in here, so you don't have to worry about me putting up slides of synthesis over and over again. But this interesting molecule here was a good starting point with a biochemical potency of approximately 12 micromolar. The SGC was able to solve an X-ray structure of this bound to DCAF1. And that gave us a really good idea of how these things bound to the protein of interest, and what changes we might like to make chemically to improve upon that binding and make a better, more potent drug. So that's what we set out to do. Here's the compound from the previous page shown up here in the top left. An analysis of the X-ray structure that was obtained by the SGC first showed us two things. One is the enantiomer, the preferred enantiomer was actually the other one. You can see here on the left, this aromatic ring with the chlorine is pointing out towards us. If you flip that away and push it into the background, and we did some modifications and optimizations of these two aromatic rings, we go from 12 micromolar to 100 nanomolar. And this was OICR 8268, which was the first compound that we published on. And shown here again is a nice X-ray structure showing how it binds into DCAF1. 8268 was a very good potent compound biochemically, but it didn't work very well in cells. So we had to figure out a way to improve the physical properties and more drug-like properties of this molecule, which got us focusing on the central part of the molecule, this pyrazole structure here. And we wanted to also, knowing how it binds, we wanted to branch out from this chemical structure out into the solvent front, into the physical space where VPR binds. We figure it would be a better drug if we could have something in the space that VPR wanted to occupy, as opposed to having empty space there where VPR could just essentially bind over the top. But what we found was if we just alkylate that to put a substituted chain on the pyrazole directly, it killed the activity. But what we were able to do was to switch this pyrazole to a pyrrole. And you can see it's not optimal. We take a bit of a hit in potency here. But then when we substitute on the carbon next to that NH, we do two things. We dramatically improve the potency, but because we maintain this H bond that we kind of knew was forbidden, so binding to the carbon here instead of to the nitrogen was the way that we were successful in developing a 2 nanomolar inhibitor of DCAF1. Here's a summary of the this is kind of the report card, I guess, if you will, of OICR 41103 as a standalone small molecule binder of DCAF1. You can see here against the panel its criteria that we were aiming for. It checks all the boxes. It was very potent biochemically. It worked in cell assays, in 2 different cell assays, either the nanobret or the thermal stability CETSA assay. It was not toxic in a general sense, up to 10 micromolar, and we had great selectivity across a small panel of other WDs that we tested against. The enantiomer also, we were able to make the mirror image of this compound, and that provides a nice negative control for testing the effects of this compound in further assays. Okay, so we were able to develop small molecule inhibitors, quite nice ones. And now to kind of tie together to what I was talking about protein regulation, I want to talk a little about what is a new type of concept in drug design. And instead of blocking a given protein's function with a drug in a traditional lock and key type schematic, if you want, in your mind, would it be possible to utilize the UPS system? So instead of just trying to put the key into the lock, what if you can just take the lock kind of right off the door and remove the protein from in the cell and then essentially reduce its function that way? So that's what's kind of shown here on the right. Across the top of this panel, you have the UPS functioning as it normally does, where it has an endogenous substrate that fits nicely into this pocket, and then the ubiquitin gets transferred, and then it gets sent to the proteasome. But what if you had a binder to that E3, either in that pocket or in another pocket, and you had it with a tether or a chain to something that binds another protein that you're interested in? And so instead of having the endogenous substrate dock into the E3, you could essentially put in whatever protein you want that you have a binder for. And then that's going to get the ubiquitins and get sent to the proteasome, and then you can degrade that protein instead. So using PROTACs to hijack the cell's proteomic machinery to initiate the targeted protein degradation of a very specific protein is something that a lot of people are studying these days. Thousands of them have been reported, at least preclinically. The vast majority of them rely on only 2 E3 ligases, the VHL or the von Hippel-Lindau protein, or cereblon. There are currently 90 PROTACs that are being investigated in clinical trials, many with implications in cancer, including ARV-471, which I believe had its NDA filed this past year or within the past year. And it will be looking for approvals of these maybe in 2026 or 2027. ARV-471 is a degrader of the estrogen receptor. and it's being studied in phase 3 clinical trials for ER-positive, HER2-negative breast cancer. One thing that is of a high concern is the focus on VHL and cereblon and the potential implications that has for resistance of targeting cancer cells in this way, developing. And that's been shown, at least in cells, to be somewhat of a concern that when they do try to force mutations, the mutations do seem to arise on the E3 side and not on the protein of interest side. So it opens up a field of we need to find other tools in the toolbox, as they say, to maybe overcome this in the clinic if need be. And we thought that is going to be a great opportunity for DCAF. But first, WDR5 is an essential gene in the oncogenic process. It's a scaffolding protein for the complex. It's an epigenetic target that's involved in the methylation of histone 3 at lysine 4. This mark is associated with proliferation. And we have a lot of history working with WDR5. We had a lot of assays set up to study the impacts of WDR5, so we thought this would be a good starting place to work on PROTACs. A very simple slide to show how naive we were when we started. We figured, oh, we've got a good DCAF binder and we've got a good WDR finder. We'll just staple them together with some really simplistic things and we'll have degraders and everything will be great. And it didn't quite work out that way. It's a very empirical undertaking. We synthesized more than 100 of these bifunctional molecules, and the synthesis is many steps and quite complex. We ended up having to go all the way up to a linker that had 9 units of PEG in it to be able to get reliable functional PROTACs with good degradation and good potency. And there's the bottom point here. The linker between the two functionals is very important and should not be overlooked. But at the end of the day, we were able to make quite good PROTACs. Shown here on the right is an X-ray structure of one of them, OICR 4114, bound to both DCAF on the bottom and WDR5 on the top. You can see here compared to the negative control, 4114 degrades WDR5. It's about 50 nanomolar EC50 with a Dmax of 75%, which means it removes approximately 75% of the WDR5 protein from the cell. OICR 4114 kills MV411 cells, as you would expect for something that we know is a cell line that's quite sensitive to WDR5 inhibition, so that fits in there. But unfortunately, this compound here shown on the right, you can see it has the same piece on this side. That's the WDR5 binder. And this is a direct comparison kind of our DCAF piece to a VHL piece. And in this case, as shown in blue, the VHL one wins out. With this combination of the protein of interest in this specific cell line, DCAF one is not as good at degrading WDR5 as the VHL analog is. And so with that, all that's left to do is some quick acknowledgements. There's a large team of chemists and biologists at OICR and a lot of biologists and structural X-ray structure people at the SGC, along with our collaborators at Pyramel who provided some chemical support, the people here that were involved in the Del-ML screening, and a couple people at Merck, Tobias and Paul, who prepared a tracer that we were using for one of our cell biology assays. And that's all for me today. Looking forward to questions in the discussion.
Okay, moving on to our last speaker, Dr. Richard Marcellus, who is currently a senior scientific advisor in the drug discovery group. And Richard joined us after having spent over a decade in industry. And he spent considerable time at Geminex Pharmaceuticals in Montreal. Richard has quite a diverse background. His expertise spans many aspects of the drug discovery process, including protein production and characterization, assay development, and compound screening, and worked on validating targets, and as well as now he's heading the entire biology group at OICR. So with that, turn it over to Richard.
Thanks, Rima, for the introduction. I'm just going to present some data we've been putting together on WDR5, looking for new applications, how to best use this asset. Well, I'm the one presenting. Joe, Sam, and Quan, kind of the bioinformatics team here within drug discovery, actually Quan's also with AO, have been doing a lot of the work. I'm just the messenger. Okay, so Brian gave you some introduction on WDR5. I'll just go over some of the key points. So basically, it's an epigenetic enhancer that is promoting gene transcription from active promoters. So it does that by interacting with two different complexes. There's the MLL compass complex. That's the one that's been best characterized. And a lot of the literature on WDR5 concerns MLL1 rearrangements and leukemias, but it also is involved with the NSL complex. So both of them target two different histones, and they're both positive marks for gene transcription. In addition to the epigenetic role, WDR5 also stabilizes MYC on some promoters, particularly promoters where MYC is a bit weaker, and so it's driving in a more immediate sense MYC transcription. So all of this helps with proliferation, helps with cell growth. So targeting that, disrupting these complexes, its scaffolding function within these complexes would be expected to have a role in shutting down growth in cancer cells. So as I mentioned, our initial focus at the very beginning of this project, when we started with the SGC, they brought this target to us early on, and it was around MLL rearranged AML. So, and that path did look good. We got great inhibitors, as Brian has described a bit, and really specific stuff. But it turned out we did partner this project initially with Celgene, but through the more advanced preclinical work, it looked like the path to the clinic would be quite difficult. The therapeutic window was a bit slim, and in that patient population, we just couldn't, or at least Celgene didn't think it was advisable to go forward into it with this asset. So we have pivoted a bit and looking for solid tumor indications that could help advance the asset. We had done some studies earlier on, some cell panel work, and we saw that some solid tumor indications were sensitive, at least subsets within them. We didn't have the data quality, though, to really nail that down and figure out where we should go forward with it. More recently, the Broad Institute has put together a screening platform called PRISM, and I'm going to describe that. And we've submitted five WDR5 inhibitors have been sent for this PRISM analysis. 12256 is our most advanced candidate. That is the one we've done a lot of preclinical work on in animals and toxicity studies. We have 14309, which is very similar to 256 structurally, but it is 5 or 10 times more potent. So it's a really nice potent compound, just a little bit less advanced in profiling than 256. I've also included two other compounds, 13583 and 2883, have quite different pharmacokinetics. The way they distribute in the animal, quite different. They're much more lipophilic, less polar than 256 and 309, so they distribute much more broadly if you put it in an animal. The structures are moderately different. They are based on the 256 scaffold, but there's quite a few differences. And then we have 40499. This is a competitor compound that was produced by Stephen Fezzik in collaboration with the Tennessee labs down in Nashville. It's completely unrelated structurally. It's a good, high-quality compound. They published a lot on it, so we included it just to help us help us decide between on and off-target activities. So the goal was is to figure out, okay, what other tumor indications could be promising? And also because the Broad has so much bioinformatics on the cell lines that are used, we'd be able to look for biomarkers of sensitivity with the data set. Okay, so this is how the Broad screen works. Normally, in the past, when we wanted to do multi-cell line screens, you'd be testing your cell lines one by one, and that is a lot of work. What they have done instead is taken 950 cell lines, basically most of the stuff that was in their CCLE cell line collection, and they put unique barcodes into each cell line. And then you can assay the cell lines in mixtures. So they've made 40 pools out of these 950 lines, and the pools are matched for similar growth rates. So that way they can do 40 dose response curves with your compound rather than 950 dose response curves. And then the readout is done by DNA sequencing. So they plate the pool of cells, they can sequence at the beginning and then sequence after it had been exposed to your dose response curve or compared to the vehicle controls. So they can see which are dropping out, and the ones that are dropping out in the sequencing or disappearing are the cells that are getting killed. So they've also, all of these 950 cell lines, the RNA and DNA sequencing's been done on them, proteomics. They've got their DepMap platform where they've got CRISPR and RNAi genetic knockdowns of all the targets. It's all on the same cell lines. And they've also run thousands of drugs and other people's compounds through this whole panel. So there's a huge wealth of information you can get from this. The way they run the assay is it's a standard dose response. They use an 8-point curve, and they do it in triplicate, everything from 10 micromolar down to 4.5 nanomolar, 5-day treatment, and then, as I said, read it out by sequencing, and then you can access all your data via a web portal they've set up. And you can also download all the data, which we have also done. Okay, so this is just a big plot of the 950 cell lines with 12256, our main compound, running across them. So that's the data they're giving you, all of these kill curves. And then they also calculate the area under the curve for you. So basically the area under these curves is the number of cells that have survived the treatment. The area above the curve, which sometimes we use as well, is the surviving cell population. This is focused down into just the leukemia lines. As I said, 256 was initially intended, or our WDR5 inhibitors were initially intended to be used in leukemia. These are, I've highlighted in thicker lines, are two workhorse lines that we used extensively. So MV411 are very sensitive. That's this first line coming down, 40 nanomolar IC50. That lines up quite well with what we were getting internally. THP is the line we used to always use as our negative control. And you can see it's sitting very resistant until you get to high concentrations. And then the whole spectrum of other leukemias that are going in between. So there's a broad range of sensitivities. For most of the analysis you're seeing in this presentation, I'm using the area under the curve as the metric rather than the IC50s of these sigmoidal fits. Okay, so here is a sensitivity. These are box plots across all the different tissues, or at least all the different tissues that had a reasonable number of cell lines. So the myeloid, so that's your leukemia. Lymphoids, we'd also known were quite sensitive, so that's our B-cell lymphomas and things. So the myeloid and the lymphoid are quite sensitive. Here we've ranked them from lowest average sensitivity to highest average sensitivity. But even though myeloid and lymphoid don't have the highest average sensitivities, the most sensitive lines are generally found in those lineages. So that's where our MV411 is going to sit down here. Lymphoid also has some nice sensitive lines. The other one that's showing up, and is one we had known about before, is the CNS brain tumor lines. There's some really sensitive lines among them too. Across the other tumor types, you can see there's big spreads between more versus less sensitive cell lines. So the spread is very high, but the average sensitivity from the most sensitive cell grouping to the least sensitive cell, it's not that dramatic. So you can see that within every tumor type, there generally are some fairly sensitive lines as well as fairly resistant lines. So it's not a broad killer that just kills everything. They're within the tissue types, really, really broad spread. And we were hopeful that that broad spread would help us identify some biomarkers. Okay, so within the Broad's PRISM data portal, they do allow you to do a little bit of data mining. The not the disadvantage, but the limitation of their portal is you can just analyze all the tissues or all the cell types as one group. So what we've got here, these 3 volcano plots are looking at gene expression correlations. So what genes are expressed high in your sensitive cells? That would be this group here in the top right. What genes are expressed high in your resistant cells? And that would be the top left here. And then the same thing with CRISPR. You can get the RNAi data sets. And the repurposing, what these are all the drugs or probe compounds that they've run through their panel. And there's thousands of those, I think. So you can see, are there any other drugs, probes that have a similar killing fingerprint to the new agent you are testing? So what you can see here with 256, first of all, your correlation coefficients are not particularly high, around 0.2. Also, the stat values, the statistical significance, not particularly high. And I'll show you something where it is high to give you a bit of a reference. So what it showed up here, I just clicked on the top hits in each of these, and then the list is included down below. It's kind of a random list. The drug that was most similar, not particularly similar, but at least the most similar, was just a multi-targeted kinase inhibitor. Then there's random mitochondrial proteins showing up among the gene expression and the CRISPR. Not highly satisfying stats on this, and also not something we would know from the biology of WDR5. Doesn't necessarily make a lot of sense to us. Okay, so I'll move on. This is an example of our DCAF1. Now, this is not our probe that Brian had showed you. Before we had selected the probe, we had some other compounds that had been looking quite good that were actually fairly cytotoxic, and we sent 2 of them for PRISM analysis. And here is one of those. So 2 went there. They both show the same pattern. And here you can see on the gene expression, we have a really high aligning point up there. CRISPR again, and you can see the stats. We're up above 10 to the power of 15. They're really strong stats. So we got a CRISPR knockout, we got a gene expression, and we got a couple of really, really strong hits on the repurposing compounds. And they all aligned on one thing, which is a gene called SLC16A. It's a monocarboxylate transporter. Brings in things from outside the cell to inside the cell. So it turned out we actually had an off-target activity we were not aware of in these pre-probes that is targeting quite effectively this target that is clinically viable, like those drugs you're seeing in the top corner from Bayer, and they are good compounds. They're intended for the clinic. They really lined up really well. However, in the process of finally deciding on a probe for decaf, we completely lost this activity. So it's just showing you, if you've got a really focused target that is a kind of a simple phenotypic readout or a simple consequence, you can see really strong hits. But that's not what we're seeing with WDR5. It's a bit more broad in its activity, given that it's sort of shutting down gene transcription, not simply hitting a transporter of a nutrient. The other thing that we saw looking through the Broad, again, this is just Broad analysis. So when I took the top hits, the top 100 hits from each of our WDR5 inhibitors, so OICR, there was 4 of them, tried to overlap them to see which hits were in common. Mostly we were not getting common hits. I think we're just seeing noise. Just a few hits showed overlap. When we took our best 3 compounds, the most closely aligned of our 3 compounds, and then added in our competitor's WDR5 inhibitor, we did a little bit better, but you're still seeing there's a lot of individual hits. When I took this group of 26 hits that we were seeing shared among the 4 compounds, and we dumped that into some AI analysis. There was no statistical significant enrichment for any specific biological pathways or processes, and that's coming from things like reactome, and no protein-protein interactions were seen. So I think by trying to look for markers for sensitivity across all tumor types, we realized was probably not going to be successful. The reason a cell might care about WDR5 could definitely differ between cancer types. And so we felt the best way to go is to do everything one tissue type at a time. So this is Sam, who you just met earlier. Sam and Quan downloaded all of the dose response data, area under the curve, from the PRISM screen. And then the Broad also makes available all of the gene expression data, the CRISPR, the repurposing compounds, all of that is freely downloadable. So we downloaded it all. Then we looked at doing comparisons between the compounds. We wanted to focus on target correlations. The risk here is any small molecule could do more than one thing. Of course, we designed it to inhibit one target, but it could bind to other things as well that might contribute to a phenotype, a killing phenotype or some other kind of phenotype. So we felt by aligning compounds, especially ones that showed structural differences, could really help us focus in on the things that were most likely to be relevant. So it basically reduced the noise. There's 20,000 genes that are going to be randomly expressed across these 900 cell lines. There's a lot of patterns that could emerge that have got nothing really to do with the biology of your killing. They're just noise. And also, we didn't want to be focusing in on off-target correlations. We wanted to focus on our mechanism. So that was able to be done. And then with the biomarker identification that Sam and Quan have been doing, just looking to see, okay, which genes are showing positive versus negative correlations. As I said, at the PRISM, we could only look at the whole set as a single entity. But once we had everything downloaded, we could then focus on single lineages, which is what we've been doing. And now moving forward, we want to go into even the subtypes within the lineages to try and hone in on the most relevant biomarkers. We are looking generally for hits where at least 3 out of the 5 compounds are sharing the gene biomarker. And then categorizing these lists by using reactome and also using LLM type analysis. They're getting pretty good actually at taking gene lists and telling you what they see in your gene list. And then comparing the hit pathways across different tissues. Are we killing different tumor types for different reasons, or can we see some things in common? Okay, so first of all, when we compared the killing fingerprint of each of these compounds, we saw that our main compound 256 was correlating really well with the most potent derivative of 256, our 309. So that's one of our best correlations. Also, the compound from Stephen Fezzik was a really tight correlation. So these are our 3 tightest correlating compounds, which is nice because 499 is structurally completely unrelated, while these two are quite similar. The ones that are a bit more structurally different from 256, but still related to it, we're showing decent correlations, but definitely, especially with 13583, starting to drift a bit and showing some different patterns. So we have been doing some analysis where we're looking at the overlap between all 5, but the most recent work, we're focusing in on things that are shared between the 3 tightest compounds. So this is an example of what we've seen in many tissues. So this is a Venn diagram where we're looking at all of the gene expression correlations for the individual drugs and then seeing how they intersect. This is an example of colon cancer, bowel tumors. So you can see here at the outside, all the color from this sort of heat map is showing up in the outside. We're seeing a whole bunch of correlates that are single hitters found in only one compound. And we would expect that we should be getting a lot of overlap. These compounds are functionally doing the same thing. Yet we see nothing in the middle. What we're focused on are genes that are in common. And we're not seeing anything at all that's shared between all five. Even when you go to 4 out of 5 or 3 out of 5, we're really not seeing much in the positively correlated genes. And the negatively correlated genes, again, it's the same sort of thing. We're getting very, very little overlap. This pattern we did find with quite a few different tissue types. So I just shown you the bowel, zoomed in. Here it is from a 50,000 foot view. There's many cancer types, uterus, pancreas, where we're not seeing positive or negatively correlated genes. There's a couple cell types down at the bottom where we're seeing a small number of genes that correlate and are shared across up to shared across all five. But generally speaking, we're not getting a lot of overlap in these tissues. Where we did have better success was in the myeloid. So this was the whole WDR5 program was built out of a lot of literature strength in the myeloid leukemia space. And here we were seeing a lot of overlapping genes, particularly in the negatively correlated. So that means genes that are expressed at high levels in the resistant cells and at low levels in the sensitive cells. We saw a little bit of positive correlating genes, but a lot less than the negatively correlating. With CNS, so brain tumors, we're seeing reasonable number of negatively correlated, but we didn't see any positively correlated genes. In contrast, liver didn't show us any negatively correlated genes, but it showed us a lot. We got a bit over 100 positively correlated genes. We haven't had the chance to mine this data set yet, but it's sort of interesting how it stands out as being quite different from our myeloid and CNS. And then in bone, we've got a little bit of both. Not so many genes, but we're seeing both positive and negative. Now, one thing we've been doing, and this is work that Joe's been doing in my group, is just taking these big lists of genes and dumping them into Reactome. And the Reactome will light up all the different pathways. They've annotated about 8,000 genes. So we'll take our list, dump them in, and then there's sort of a nice visual here where you can see in yellow, kind of a yellow heat map, where your genes are mapping. And these, what I'm showing you here are things that are considered statistically significant. So Reactome is seeing more than one example. They're seeing a couple of examples within these highlighted pathways. If you released that statistical test and just asked every gene in your list, it would light up most of this. So we see stuff scattered all over. But when we focus on the things that are most likely to be relevant based on the stats, we're just seeing a small grouping of things. And what was interesting here is, for the most part, we are not seeing much commonalities between the different tumor types. However, between myeloid and CNS, we were seeing some things showing up. And notch signaling was what Joe had homed into most. Notch is also showing up in the lymphoid analysis. So notch signaling, or at least the genes involved in that notch signaling, are seen to be more highly expressed in the resistant myeloid, resistant CNS, and also resistant lymphoid. This is just zooming in a little bit, showing the kind of pathways we're seeing. None of this is, or not, I shouldn't say none of it. Some of it is not things that we would have known anything about, like this MITF factor. We're seeing that showing up. And we're also seeing, we are seeing things in gene expression showing up. We're seeing our notch showing up, notches in this area, signaling by notch, and there's other things. So you can see by comparing between the top and the bottom is our myeloid versus our CNS. You can see some similar patterns. So it's sort of interesting that you wouldn't have thought of brain tumors as being at all similar to myeloid, but we are seeing patterns that are the same. Interestingly, though, the gene lists themselves are not the same. It's just the pathways. We have representative genes from these pathways, but the actual gene itself is differing. Different isoforms, different parts of the pathway are what are overlapping. And Reactome mining can show us that. But if we were just directly comparing the gene list, we wouldn't see anything, or see very little, actually. And this is, as I said, we had been focused on trying to get hits that were shared by all 5 compounds since Most recently, we've been focusing down on what we think are our best closely aligned compounds. So two of ours and then one from our competitor. And we can get a little bigger gene list when we do this. So myeloid, we can get up to 180 genes and CNS down to 57. We're still not seeing much in the positive correlation. So myeloid now we're up to around 11 hits. So it's maybe enough to look at the biology behind them. So far, the analysis has just been done out of the 5 out of 5 or 4 to 5 hitters. The other thing we've been doing is just taking these lists, as I said, dumping them into an AI based LLM and seeing, okay, what can they tell us about these gene lists? We don't tell them much information. We just say, here's a gene list. What do you see? And this is what we're seeing. So between myeloid and GBM, we had, that's our brain cancers. As I said, the gene lists were quite different. There was only one thing shared of all these genes, and which was argonaute, which is involved in RNA silencing. Not something that was obvious to us that should show up, but it was there. The notch signaling was quite strong in the GBM samples, but there's also things like RNA regulation, mitochondria things. In myeloid, we're seeing DNA replication. Again, these are things we're seeing in the resistant cell lines, not the sensitive cell lines. And we're seeing some proliferation links. So this helps us categorize things. We're sort of thinking of it as a complement to Reactome. Reactome is limited to the genes that have been annotated. This will go and mine things like your gene cards and things and pull out information. So it'll give you some idea of how strong of a link it's seeing. So that's what we're getting at. We're trying to basically take these gene lists, find the stuff that would be biologically relevant, not just marker genes, but it's also to know marker genes that are in the pathways that we'd expect to be modulated by WDR5 inhibitors. Now, here's something Sam has just taken some just early steps at trying to figure out sensitivity signatures. So what she did here was took the top 10 negatively correlated genes in myeloid and the top 10 positively correlated genes, and then she averaged them. And this is the kind of spread you're getting. So this is 1 over AUC. So what this is, so the cells that are dying are the ones that are in the top corners. So what we're seeing here on the left is these genes are quite so sensitive to WDR5 inhibition and are expressing at low levels. This is the TPM, is the expression level of the genes averaged out over the 10. So here we got low expression of these 10 marker genes, and they're very sensitive. And then here we've got marker gene sets that are highly expressed in our sensitive group. So eventually we want to be able to put together some gene signatures that will combine both the highly expressed genes and low expressed genes to come up with something that's a fairly robust signature. The other thing we want to do is, rather than getting tricked by markers that are simply telling you about different subtypes within a lineage, we're going to drill down into each individual subtype. So on the left side, the first two are our AML and then some other myeloproliferative neoplasms. So normally when we look at myeloid, these are grouped together, but clinically they're different. If we were interested in myeloid, and that you can see the myeloid, the AML are the most sensitive, much less so. So by focusing in on the 35 cell lines that are AML, we might be able to pull out biomarkers that are a bit more specific to the biology without being contaminated by these other lines. With brain cancer, we're also seeing some differences. The embryonal tumors will likely be quite different from the diffuse, the gliomas, the GBMs. So being able to analyze these separately will be helpful. In the lymphoid, we're seeing that our most sensitive group is in the mature B cell neoplasms. So again, we haven't done that much mining of the lymphoid yet, but I think it's going to make sense to focus in on the most sensitive group and take out the contamination by the other subtypes, which could have marker genes that could show up as resistance markers, but really they're just markers of this subtype that doesn't happen to be very sensitive. So that is something we're going to move to next. The downside of this is as you reduce your cell number, your statistical power is going to drop. So there's a kind of a sweet spot there. You don't want to get too small of a group or you won't be able to see anything. So the next steps with this project, we're continuing the data mining, trying to understand these sensitivity and resistant patterns we're seeing. Also, as I said, we want to focus in on subtypes within the sensitive lineages to avoid any subtype specific marker genes, and then ultimately create gene set signatures that we could allow that could create predictions. We could then sort of look through the primary patient sequencing data at the TCGA and see, okay, how common are these signatures? Because remember, these signatures right now are coming out of cancer cell lines that people grow in plastic dishes. Ultimately, we want to be able to treat primary cancers. So we want to see, okay, if we get a signature, let's see if the signature in real patient data reflects what we're seeing in the tissue culture. And also look at correlations to drug repurposing data. There's lots and lots of drugs there. So what drugs do WDR5 look like, and which drugs look the opposite of WDR5? For example, in the GBM space, people are developing Notch inhibitors. We're not killing the cells that are high in Notch. We seem to be killing the ones low in Notch. So maybe there's potential for combinations that could be revealed by comparing that data with ours. And also just looking at standard of care within the sensitive cancer subtypes, just to see, okay, how do we compare with sensitive standard of care? Could we complement it, or are we doing something very similar to the existing treatments? So that's where we're standing. It's been a big job. Spent more than a month with a couple of people working on this pretty hard. So there's a lot of data there. The spreadsheets we're playing with have 500,000 rows on them. They're really big, big data sets. And it's been interesting, but it's definitely been harder than we thought it was going to be. And thank you very much.